Welcome back, everyone. Today we are wrapping up our discussion of enzyme mechanisms with a few more examples. Learning outcomes from today, we are continuing to learn how our groups can perform catalysis and the types of catalysis that we are seeing. We're seeing a lot of acid-base catalysis and some covalent catalysis as well. Today, we will see how cofactors can participate in the catalysis with a mechanism from carbonic anhydrase. Finally, we are going to kind of pull all of what we have learned together today and compare and contrast the many ways enzymes behave as catalysts. Today's Meet a Scientist moment is Kelly Chacon, who uses she, they pronouns. She is an associate professor of chemistry at Reed College. They are of white and Mexican descent. And they study metal ion transfer between proteins. So today is going to be the first time we really have a clear example of an enzyme that uses a metal ion to perform catalysis. And Kelly studies how metal ions are put into proteins, are transferred from one protein to the next. Cool stuff. So we are continuing our journey of learning about covalent catalysis today. We saw an example of this with serine proteases, but we're going to give it a bit more focus today. Sometimes covalent catalysis is called nucleophilic catalysis because there's some type of nucleophilic attack that's often performed when we're forming a covalent bond, right? This nucleophile is most often from the catalyst, and the electrophile is most often from the substrate. Here are some examples of nucleophiles that we would expect to see in biochemistry. So very common nucleophiles are going to look like hydroxyl groups, right? Or thiol. The OH and SH are more or less the same when it comes to the type of chemistry we'd expect. Of course, we should recognize that the nucleophilic form, though, is O-. It's a much stronger nucleophile than the OH. Same with that thiol. But it's not just this hydroxyl group or thiol that can do a nucleophilic attack. We, of course, can have the lone pair from amino groups or a lone pair on a nitrogen within an imidazole group that we might see on a nucleophile like histidine. So nucleophiles that you'd expect from organic chemistry, again, we definitely see these in biochemistry, particularly OH, SH, and NH. The most common electrophile that we are going to see in biochemistry is going to be the thing that looks like a carbonyl carbon. Either it's a carbonyl, maybe it's an amine, maybe it's a metal ion. But for the sake of this class, really things that look like carbonyl carbons. What are some amino acids that we would expect to see performing this type of covalent catalysis? Well, histidine, cysteine, lysine, aspartate, and serine are reasonably nucleophilic and can form that transient covalent bond that then will be broken at the end of the mechanism and leave as a leaving group. Later in this class, we'll see examples of cofactors that also perform the same type of reaction: thymine pyrophosphate, pyridoxal phosphate. Don't worry too much about that now, but no, cofactors also do this type of chemistry. The first example I'm going to show of this type of covalent catalysis today is an example where we're stabilizing an intermediate in the decarboxylation of acetoacetate. So if anyone's done the keto diet for any period of time, your body was definitely performing this reaction. But likely this reaction is happening in all of our bodies, especially if we've done any short-term fasting or exercise. And I'm not going to do the full mechanism for us here, folks. I'm just going to show us the difference in what the reactive intermediate would look like in a catalyzed reaction compared to a non-catalyzed reaction. Okay, folks. So in this reaction, what's going to happen is a decarboxylation of acetoacetate. So we can imagine that if this reaction were to be happening in a way that was not catalyzed, we'd end up with an intermediate that was charged. If we do a decarboxylation, what's going to happen is that we'll end up with an intermediate that looks like this, although we know we could and should draw a resonance structure where the negative charge is now on the atom that's more ready to hold that negative charge. So that's what our intermediate might look like. Now, what does the intermediate look like when the reaction is catalyzed by an enzyme? So when this reaction is catalyzed with an enzyme containing a nucleophilic amine, you end up with this intermediate where this nitrogen has done a nucleophilic attack. We still have this carbon arrangement like we do on the left-hand side. But what do you see in the catalyzed version compared to the uncatalyzed version? You notice on the left-hand side we have that negative charge. And yes, we know it is somewhat stabilized because we can draw that resonance structure and share the negative charge across the carbon and the oxygen. But on the right-hand side, we have entirely eliminated that charge. And so we've done this kind of quasi-charge stabilization through covalent catalysis. So this is the first example of what we're seeing. The catalyst can stabilize a reactive intermediate by eliminating the need for it to be charged at all. The second type of catalysis we're going to focus on today, we are going to see a full mechanism because I want you to see a full mechanism using a cofactor, but it's fairly simple, so I thought it was a good one for us to walk through today. We are going to see metal ion catalysis by the enzyme carbonic anhydrase. Nearly a third of all known enzymes require metal ions, so it's good for us to see an example of this today. A wide variety of metals can be used to perform catalysis. Iron in its oxidized or reduced states is going to be what we see today. Today we're going to be using zinc, but into the future we'll see iron as like the most common example. Now, these metal ions can perform catalytic functions, but they can also be structural, and this will definitely be true for many enzymes we talk about for the rest of this class. So magnesium, zinc, structural and catalytic, whereas something like manganese, iron, cobalt, those are going to be more purely catalytic in nature. Now I'm going to give us kind of a peek ahead as we begin to think about this mechanism. What are we going to see? We are going to see that this metal ion is important for binding the substrate and orienting it correctly. We are going to see in the future, but not today, how these metal ions can do a redox reaction through their own oxidation state. Think iron, Fe2+, Fe3+. Today we will see the metal ion do some electrostatic stabilization or shielding of negative charge. Something to keep in mind as well, folks. Metal ions are not pH sensitive. Last time we were together, we talked about how these R groups are going to be the best at doing acid-base catalysis when pH is near pKa. Metal ions don't have a pKa, so that is not true for them. Finally, folks, we are going to see how a metal ion can make a water molecule more acidic or more likely to give up its proton even when the reaction is happening at a physiological pH of 7.4, which is more or less neutral. So let's jump into this mechanism, folks. Let's start by drawing in the catalytic site. This catalytic site has 3 histidines that are all coordinating a zinc ion. So all 3 of these histidines are bound to a zinc ion. The enzyme carbonic anhydrase is responsible for converting CO2 released through respiration into carbonic acid. So let's bring in our substrate. But before we get to our substrate, folks, we have a second substrate of this enzyme. Which is, you guessed it, H2O. We've seen this in other mechanisms, haven't we? So the very first thing that's going to be happening in this mechanism is our zinc ion is going to polarize this water molecule. What do we mean by polarize? Well, the zinc has the 2+ charge, right? It is going to kind of pull the negatively charged, the partial negatively charged oxygen of this water towards it and make that charge slightly more extreme, right? If zinc itself is positively charged, it's going to interact with that negative charge and kind of pull on it a bit. That's what we mean by polarize the bond. It's kind of pulling more of that negative electric charge towards it. Now, when that's happening, when the OH and the partial negative on the O is being stabilized, this water is going to be more likely to give up a proton and become OH minus. Why is that? Well, let's draw the first step of this mechanism and talk more. Elsewhere in the catalytic site, folks, we have a base. I'm not going to draw the amino acid. I'm going to put just a general base, B. The first thing that happens is this base deprotonates our water molecule. That means that our base is now protonated. We'll give it a positive charge. Our catalytic zinc didn't really perform any reaction, right? But its catalytic role was to, let's draw this in now, kind of bind onto that water by grabbing onto the partial negative charge of that oxygen and polarize the bond in water. Since that O minus, that partial minus, was being stabilized by the positive charge, the water was more likely to give away that proton and become OH in this step. You see how this OH now has a full minus charge? Well, that full minus charge is electrostatically stabilized by the positive charge on the zinc. So zinc's catalytic role to bind that water, to polarize that bond, so that the water can give up its proton, become OH minus. And then that zinc stabilizes the OH minus since it is positively charged. And now we can draw in step 2, folks. Don't forget our substrate, that looks a little bit like an O, was CO2. But as we release CO2 from respiration, we don't just have gas bubbles forming in our bloodstream, we are converting CO2 to bicarbonate. OH minus is a fantastic nucleophile. So it can attack the biochemist's favorite electrophile, the thing that looks like a carbonyl carbon. And you'll see that we have reached our final destination here, folks. The zinc is the first time we've seen a metal ion cofactor performing the catalytic role in a mechanism by mostly doing some charge stabilizing. So pretty cool stuff. Now we're going to move on in the next video to discuss some additional ways that enzymes increase reaction rate and act as catalysts.